As we begin our trichology chapter, we will be going over the chemical structure of hair. We will discuss the major layers of hair, different growth patterns, and then also we will get into analyzing the hair and scalp as well. We will go over a couple common conditions of the hair and scalp to look for when analyzing a client's hair or providing a scalp analysis. The two primary parts of the hair strand are the hair roots and the hair fiber. The hair root is located below the epidermis, which is the outer layer of skin. The hair fiber projects beyond the epidermis. Hair is composed primarily of a protein called keratin. Hair strands are created through a process known as keratinization. Living cells that make up hair are formed in the hair bulb. Newly formed cells are pushed upward through the epidermis, causing them to emerge from the scalp and become fully keratinized. As hair becomes fully keratinized, it is no longer alive. Hair is mainly composed of keratin. Major elements that make up hair use the acronym CONS, COHNS, which stand for carbon, oxygen, hydrogen, nitrogen, and sulfur. Hair is an appendage of the skin, and the CONS elements can also be found in skin and nails. Protein building blocks, known as amino acids, link together end to end to create the hair strand. Amino acids are linked by a very strong chemical bond called a peptide bond. Amino acids and peptide bonds link together to form polypeptide chain. Links created by these chains are known as a helix. The portion of the hair strand considered to be alive is found below the skin and consists of the hair follicle, the hair bulb, and the dermal papilla. The hair follicle, a tube-like structure in the skin or scalp that surrounds the hair root and anchors the hair in the skin. Hair follicles will not be found on the palms of the hand or the soles of the feet. The hair bulb, which is located at the base of the follicle, has a bulb shape and contains living cells that will form the hair strand. The dermal papilla, a cone-shaped elevation at the base of the hair bulb. It contains blood vessels that supply nutrients that allow the hair to grow. Erector pili muscles are attached to the hair follicles, are also attached to the hair follicles. The erector pili muscle is an involuntary muscle in the dermis that attaches to the hair follicle. Involuntary contractions of the erector pili muscle may cause a client to experience goosebumps and may cause the hair strands to stand up on ends. Sebaceous glands are also attached to the hair follicle. They produce a substance called sebum, also known as oil. Sebum lubricates the hair and skin. The non-living portion of the hair, also known as the hair fiber or the hair shaft, consists of the cuticle, the cortex, and the medulla. The cuticle is the hair's outermost layer. The cuticle is overlapping layers of cells that protect the cortex. The cortex is the protein layer of the hair. It gives the hair strength, elasticity, and color. The cortex is made up of polypeptide chains, and the hair fiber also consists of the medulla, the innermost part of the hair strand. It's possible all clients may not have a medulla. A medulla may often be found in coarse hair, not always in fine. As the hair strands, also known as polypeptide chains, emerge from the scalp, they are linked together by side bonds. The side bonds are known as hydrogen, salts, and disulfide bonds. Side bonds contribute to the hair's strength and elasticity. A hydrogen bond is a weak side bond that can easily be broken by heat or water. It can be reformed by drying or cooling. Side bonds are also weak. They can be broken by changes in pH and reformed by balancing pH. Disulfide bonds are strong chemical side bonds. They cannot be broken by water. They can be broken by chemicals such as perms or relaxers. Disulfide bonds can be reformed with oxidation during the neutralization process. Peptide bonds, which are sometimes referred to as end bonds, are strong end bonds broken by chemical depilatories. They are not reformed and the hair does dissolve. Be mindful of products that dissolve hair when working with clients. It is not always possible to reform a disulfide bond once it has been broken. Some chemical procedures will allow the disulfide bond to be reformed, and some chemical procedures will not allow the disulfide bond to be reformed. Always refer to the manufacturer's directions. Natural pigment found in the hair, known as eumelanin or pheomelanin. Eumelanin consists of brown or black pigment, which creates brunette hair. And then we also have pheomelanin, which is yellow to red pigment and natural blonde and red shades of hair. The absence of melanin causes gray hair. Gray hair grows from the bulb in the same way that pigmented hair grows. It has the same structure, but without the melanin pigment. Client hair texture will depend a lot on genetics. Client hair can be wavy, straight, curly, coily. All of these hair textures can vary from client to client and culture to culture. And they can also be present on, multiple textures of hair on one client. A client can have really curly hair in one section and then have really straight hair in another section. You may hear the term round-celled, oval-celled, or elliptical cell. Round-cell follicles are known to produce straight hair. Oval-shaped follicles are known to produce wavy or curly hair. And then elliptical-shaped follicles are known to produce very curly hair. As you begin your hair and scalp analysis, you are checking for a healthy scalp and any conditions that may prevent you from performing your client's desired service. During the hair analysis, you will check for texture, porosity, density, elasticity, different patterns of growth that might affect your styling. You can check for common hair conditions such as, split ends, matting, different scalp conditions, psoriasis, dandruff, different head lice. You want to make sure that you don't see any kind of, ringworm or scalp conditions. Anything that, the client should be aware of or that you should be aware of will all be discovered during the scalp analysis. The hair's texture, also known as the degree of coarseness or fineness, can usually be determined by the hair's diameter. The coarser the hair, the larger the diameter, the finer the hair, the smaller the diameter of the hair. Hair density, the number of follicles per square inch, is usually classified as thin, medium, or thick. Clients will have a couple thousand hairs per square inch on average. Porosity refers to a hair's ability to absorb moisture. The condition of the cuticle can affect hair's porosity. A sealed cuticle may alter the product or processing time selected for that client due to the low porosity of the hair. Hair may have average porosity, meaning that it will process as expected, or hair may be overly porous with high porosity and require products with a lower pH to help prevent overprocessing or damage. The hair's ability to absorb will determine processing times and products used. Hair elasticity is the hair's ability to stretch and return to its original length without breaking. Wet hair can stretch up to 50% of its length and return without breaking. While analyzing the hair, you will also take note of various hair patterns that may appear. Clients may have a whorl, which kind of looks like a circular pattern in their hair. Clients may have cowlicks or hair streams that may determine the direction the hair needs to be combed when styling, or directional hair growth patterns that may cause you to reconsider the way you prep the client or the way that you execute their style. While analyzing the hair, you are also looking for various scalp conditions that may need to be addressed. Psoriasis, dandruff, dry dandruff, greasy dandruff. Each issue will need a different prescription. Inactive sebaceous glands can cause clients to have dry hair and scalp. You want to treat dry hair and scalp with moisturizing products and conditioners. Avoid products with high alcohol content. Clients with overactive sebaceous glands can have oily hair and scalp. Avoid products with a high oil content and make sure you consider a tea tree oil shampoo option, and give the client a double rinse after their conditioner to prevent buildup. As your client's hair goes through the hair growth cycles, you may notice slight changes in the hair. The anagen phase, which is the growth phase, new hair is produced during this cycle. You have the catagen phase, which is a brief transition period between hair growth and the resting phase. And then you have the telogen phase, when also known as the resting phase. During this phase of hair growth, the hair sheds or remain in place until a new anagen phase begins. All hair follicles are capable of producing various types of body hair depending on the genetics. Age, hormone may also play a role in the hair a follicle produces. From lanugo hair covering a fetus, vellus hair covering the body, or terminal hair covering the scalp. and body as well. Hair growth cycles can last anywhere from 2 to 6 years and will vary from client to client. A proper scalp analysis as well as a proper hair analysis will help you and your client have the best possible experience. It is your responsibility as a professional to do a proper consultation and scalp examination prior to performing any service.